Hello, everyone, and welcome to phyloseminar.org. The current theme is single-cell analysis, and this is the second talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Dr. Sebastian Prillo. Sebastian is a fourth-year PhD student in the Department of Electrical Engineering and Computer Science at UC Berkeley, advised by Yun Sung and Nir Yosef. Broadly speaking, Sebastian is interested in mathematical and computational biology. More specifically, he's interested in statistical phylogenetics and its intersection with single-cell biology. Recently, Sebastian has been working on branch length estimation for single-cell phylogenies and on rate matrix estimation with a focus on scalability. Welcome, Sebastian, and thank you for participating.
Thanks, Eric. I'm excited to be presenting this. This is the first time I'm presenting this publicly. I've been working on this for a while now. So this is pretty exciting. So yes, let's dive right into this. So again, the title, Scalable and Accurate Inference of Single-Cell Chronograms from CRISPR-Cas9 Lineage Tracing Data. So first I want to start by acknowledging all my collaborators throughout this and other closely related projects. So my advisors, Professor Song and Professor Yosef, and all the members of the Cassiopeia team, where we work on developing and applying all these methods for analyzing single-cell lineage tracing data. So the outline for the presentation today is, first, I'm going to introduce and explain what a single-cell chronogram is and what it represents. Then we're going to talk about how we can try to resolve these in vivo. And for that, we're going to talk about the experimental assay that we use, which is based on CRISPR-Cas9 lineage tracing. And then the focus of this talk, which is on method development. So how do we take all this lineage tracing data and use it to reconstruct or infer the single-cell chronogram underlying the cell population? And specifically in this talk, I'll be focusing on the problem of branch length estimation. Yes, here it is. And finally, I will talk about how we evaluate these methods through extensive simulations, and also how we find out in which ways they can still be improved. So let's dive right in. So what are single-cell chronograms? And by the way, I don't know if we mentioned this, but if people have questions, you can ask questions during the talk, and I will happily answer them. So what's a single-cell chronogram? So many important biological processes, such as organism development and disease progression, unfold through time, through repeated cell division. So here what you're seeing is sort of a time lapse of how cancer evolves. So time is advancing left to right. And we're starting from a single altered cell that starts dividing. And as time passes, some cells in this cancer will acquire the ability to metastasize and invade distant organs. So all these processes, we can model them with a single-cell chronogram, which is just a rooted tree, where the root represents the founding cell event, the internal nodes represent cell division events, and then leaves that are not at the current time represent cell death events, and leaves that are at the current time represent extant cells at this time. So why are single-cell chronograms important? Well, they're important because they capture the whole developmental history of the cell population. So by using them, we can try to answer all sorts of questions. And here I just want to highlight some fantastic recent work, which my collaborator Matt Jones was involved in, where they estimate these single-cell chronograms for mouse model of lung adenocarcinoma. And by leveraging these chronograms, they are able to find genes that are associated with fitness, by which I mean with proliferative capacity, and also to try and formulate a model for how the disease evolves from its initial more benign alveolar type 2-like state all the way up to the more terminal phenotype characterized by this mesenchymal state. So I want to talk about these downstream methods. The talk today will be focused on method development, but this is just to say that single-cell chronograms are really key objects in understanding the evolution of this population. So we want to estimate them. So how can we estimate these single-cell chronograms? And in vivo, and this is a tricky thing, because if we're looking at if we're looking at a population of cells growing under the microscope on the dish, this isn't necessarily all that hard because you just keep track of when cell division events happen, when cell death events happen, and by doing that, you can just map out the single cell chronogram for this population. But we want to do this in vivo. We want to do this if we're studying, if we're using a mouse model and we have induced some form of cancer and it's growing in the mice, then how can we reconstruct this from a sample harvested after we sacrifice the mice, for example? And the way that this is done, which is a very popular approach, is based on CRISPR-Cas9 enabled lineage tracing. So at a very high level, and I'll get more specific with a very concrete example on the next slide, but at the high level, the way this works is, say you're working with a cancer cell line, they're going to engineer this cancer cell line by integrating these fake genes, which called genomic DNA targets or barcodes. You integrate them into the genome, and what's going to happen is that these fake genes have very specific sequences that are designed by us that Cas9 can identify and cut to produce an inheritable insertion or deletion that's going to get propagated down the future generation of cells. So to be very specific, I'm going to show you a small example, and this is going to accompany us throughout most of this presentation. So let's take this toy single cell chronogram over 4 leaves, and let's take a look at how CRISPR-Cas9 enabled lineage tracing would work here. So at time t=0, we have a founding cell event. And for this simple example, let's assume we just have one fake gene that's going to have 3 target sites. So 3 sequences inside this fake gene that Cas9 can identify and cut. So maybe at time t=2, before the founding cell even gets to divide, maybe Cas9 is going to locate, so identify the leftmost barcode, produce a cut, and that's going to lead to an indel. So at time t=4, when the cell divides, the 2 daughter cells are going to inherit this mutation because, again, this is happening in the DNA sequence. This is why this is heritable. And both daughter cells will now have this mutation, and the process will now just continue independently over each lineage. So maybe at times t=6, we have another cut, this time on the rightmost target site, producing a different, maybe larger deletion. And so when the cell divides, now its 2 daughters will inherit these 2 mutations. Similarly, on the upper lineage, we might have another indel going on. This one's different from the one in the lower lineage. This one's blue. Maybe this is an insertion, and so on and so forth. So all of this is going on in the background as the population of cells is just growing. And by the end of the process, when we assay the cells, so if we're working with a mouse model, then we sacrifice the mouse, we harvest the cells, we harvest the cancer cells, for example, and now what we get to see is this. So what we get to see is the mutation patterns in all these fake genes in all these cells. And by using these mutation patterns, we then try to infer and reconstruct this tree and its branch lengths. And so as you, yes.
Thanks. Sorry. Can you just go back? Okay, so these red numbers are actual like calendar clock times.
Yeah, yeah, exactly.
And is this the tree on reconstructed cells, or is this the complete division history?
The tree on reconstructed cells. Are you talking about subsampling?
So for instance, let's look at 16. Let's look at the edge of this mark 16. Are there a whole bunch of cells, a bunch of cell divisions there for which we only sampled one of the lineages?
Yeah. So, okay. So far we haven't talked about that, but there's a few things to consider here, which is that in reality, we don't try to reconstruct the full single cell chronogram underlying the whole population. First of all, cell death events, those are essentially impossible to pinpoint by just looking at the current day population. So cell death events, we don't try to map those on the tree. So yes, there might have been a cell division event happening here, and then there was a cell death event, so we never observed progenitors from that lineage. Also subsampling. These tumors can be very large, right? Have billions of cells.
Okay.
And in practice, we only sequence maybe thousands of these cells. So really, if we want to be precise about what we're doing, is we're trying to estimate the single cell chronogram induced by the cells sampled at the present day. Although,
okay.
yeah.
Perfect. Yeah, the tree of reconstructed cells. And actually, do you mind just scooching, while I've interrupted you, scooching forward a little bit? I just want to, I've worked a little bit in this field, so I know what the symbology means, but and you said it clearly, but I just want to repeat it again, because it's a pretty weird, different type of data than we usually think about for phylogenetics. So if you scroll forward a little bit and just show, so if I understand correctly, the red ones are sort of sections of this artificial spliced in section that have been cut out. And the blue ones are places where you've had like an insertion coming into this artificial spliced in variant.
Yes.
Cool, thanks.
And these can be, and these can have different lengths. So maybe you get a deletion of just one base pair, or maybe 10, or maybe you get very large insertions. And these have their own distribution. So shorter indels are more likely than longer indels. So these are all aspects that, depending on how precisely you want to model this process, you're going to have to take this into account. So this is a very sort of a richly parameterized model in a way, depending that, what road you go down. And I'm going to talk about how we deal with all these things when we estimate branch lengths. But yeah, this is different from other kinds of molecular data. What else do I want to add? The only thing I want to add is that really these aspects of this caveat of looking at the chronogram induced by the sampled cells, that changes the interpretation of what things are on the tree. So originally, each edge corresponded one-to-one with the lifetime of a cell. But after we get rid of cell death events, and after we get rid of the cells that are not subsampled, now each edge corresponds to many self lifetimes potentially. So you have to be really careful when you interpret these trees in real life. But for the methods I'm going to be discussing, this is not something that is really going to matter too much. The statistical model that we're going to use doesn't care about these things, because this process is just going on in the background. Yeah, okay. So hopefully, when we talk about the method, we can revisit that if necessary. But so yes, at this point, the problem is we have these barcodes, these fake genes with these large insertions or deletions, and we want to use them to estimate the topology underlying the cell population, its history, and also estimate branch lengths. So step 1, topology reconstruction. So there is this work that my collaborators, Matt Jones and Alex Kudaverdian, and the Joseph Lab, as well as other folk in other labs, designed. We have this package called Cassiopeia, and again, we're called the Cassiopeia team too. So they worked on this before I joined, and they developed this package together with several algorithms to infer these topologies based on things like integer linear programming and heuristics such as maximum parsimony. So the way this works is you take in your lineage tracing data, which we summarize in what's called a character matrix. So this thing on the left you see here, this is from one of the main figures from this paper. The character matrix is going to have one row for each cell, one column for each character or target site. And here colors represent different indels. And if you run it through some of these algorithms, what you're going to get on the other side is a tree topology, as well as a mapping of the mutations onto edges of this tree. So in our toy example, to be very, very concrete, So if you feed in these 4 cells, then on the other side, you're going to get this. So it's a tree topology. So we know nothing about branch lengths at this point. It's a tree topology, and we also have annotations for the putative ancestral states of these cells. So now is when branch length estimation comes in. So now that we've estimated these topologies, we want to estimate the branch lengths. And how do we do that? And now, yes, this is going to be the core part of this presentation, which is the methods that I have been working on. So branch length estimation for these trees. And the way we do it, it's really simple. So we're just gonna model the process of CRISPR-Cas9 coding, and we're gonna model it with a memoryless process that happens with some unknown mutation rate of R. And we're going to assume that different sites get cut independently and get cut with the same rate R. So here, what you're seeing, this is just the density of an exponential distribution with different parameters. And down here, what you're seeing are 3 barcodes, sorry, or 3 target sites in our little toy example. And so the amount of time that elapses between the start of the experiment and the time that a target site gets cut is exponentially distributed with this parameter under IAD. Okay, so if you do this, then coming back to our toy example, if you want to fit the branch lengths, then this is what the MLE is going to look like. So down here on the left, this is exactly the optimization problem you get. And this is nice because it's a convex optimization problem. And also because these branch lengths do not depend on the probabilities of each indel occurring. And so, this is, yes.
So just to make sure, I think what's going on is it's not that you have choose like 2 cut sites and then, or something. It's like you choose 1 site and then it doesn't matter how long it is. It's just like, it just gets cut out.
You mean here?
Yeah.
Yeah. So this is modeling the waiting time, so the holding time to a cut occurring. And what I've described so far, of course, there's one more step after, which is, okay, Cas9 cuts, and now an indel happens. And how does that indel happen? Well, different indels have different probabilities, and we do not model this. And in this method, we do not model it because this does not affect the branch length estimates, provided that we know the ancestral states.
Thanks.
And we're going to talk more about that because that's one of the most interesting aspects of this problem, which is how do you deal with the unobserved ancestral states? And depending on how you do it, you might run into some issues. But we'll talk more about that later. Okay. So coming back to this, so this is a convex optimization problem. One small caveat here is that branch lengths and mutation rate are not identifiable because you can scale the mutation rate up by some constant, and you can scale the branch lengths down by that same constant, and you're going to get the exact same likelihood. So in practice, we just assume that the mutation rate is 1, and then we estimate branch lengths. And if we want to, if we know the length of the experiment, we can then just rescale the tree on the right. Okay, so this is the MLE with known ancestral states. Everything looks very nice. Let's apply it to our toy example. So if you apply it to the toy example, you're going to get this. And okay, it looks kind of reasonable, except that you're going to see that on this part of the tree, these 2 branches were estimated to have a length of 0. And let me show you a bigger example on a larger simulated tree. So here you're seeing a simulated tree over 400 cells. And if you fit the MLE on lineage tracing data simulated on top of this tree, you're going to get something that looks like this. And so what you're going to observe is that there's many parts of the tree where branch lengths are estimated as 0. So let me just toggle back and forth a few times so you can see this. You see this is happening in many parts of the tree. And this is a very common thing we see across a lot of trees, and it makes sense because if you If there's no mutations mapped on the edge, then it's pretty easy for the MLE to just greedily decide that it wants to make that branch length 0. Right. And for some of these phenomena, we can prove it mathematically. It's a pretty general phenomena. So how do we deal with it? So to address these robustness or stability issues of the MLE, we regularize it, and we propose 2 very simple ways of doing this. The first of which is imposing a minimum branch length. And this is straightforward because this just involves adding another linear constraint into the convex optimization problem. Specifically, we add a lower bound on each branch length in terms of the total height of the tree. This is still a convex optimization problem. And in this toy example, if we choose a minimum branch length of 0.3, so 30% of the total height of the tree, then you're going to observe something like this. This is a really large minimum branch length, of course. This is for illustrative purposes. But if we apply it on our larger simulated data with a minimum branch length of just 1%, you're going to get something like this. And now you're going to see that locally, the tree is now making a lot more sense. So again, if I toggle back and forth, so these 0 length edges are now gone, and it's looking more reasonable. So depending on what the downstream applications are, this might or might not matter to you, but generally, I think having local parts of the tree that are more realistic is a good thing to strive for. So another way to regularize the MLE, which is also really, really simple, is to add pseudocounts to the data. And I won't go into the math, but it's really simple and intuitive, which is we just add fake mutations and non-mutations on every edge of the tree. So if, again, this is a grand truth tree, and you run the MLE using pseudocounts, specifically 0.5 pseudo mutations and 0.5 pseudo non-mutations, then again, you're going to see that all these edges that were previously 0 are no longer 0 because we have these fake mutations that force the edge to have a non-zero length. So again, just toggling back and forth, you see that this effect is now gone. And in this case, it seems like it's a little bit stronger than for the minimum branch length, but these are all parameters that one can play with. So okay, so these are 2 really simple ways of regularizing the MLE. But now I want to talk about what I think is the most interesting aspect of this problem and the trickiest to deal with, which is missing data and unobserved ancestral states. So missing data. So yes, CRISPR-Cas9 enabled lineage tracing has missing data, and quite a bit. And this comes from 2 sources. So the first one is heritable epigenetic silencing. And in this toy example, I'm just silencing one target site in this barcode. But in practice, because this barcode functions as a whole fake gene, the whole barcode is usually silenced. And once this happens, all the daughter cells, all the progenitors are going to, all the daughter cells are going to have this this silenced barcode. So we won't get to sequence it. And the other problem is RNA sequencing dropouts. So when we sequence this data, so we sequence it through the transcriptome, and again, because of the low sensitivity, we might miss some of these barcodes. And in that case, they're just missing. So what happens when there's missing data? So, well, this used to be the MLE when we knew the ancestral states. Now, if we have missing data, what we do is we just assume that the missing data mechanism is ignorable. And more specifically, we assume that missing data is missing always completely at random. And if you do that, then the MLE just changes by removing any term that involves missing data. So it has this exact same functional form. It's still a convex optimization problem. It still does not depend on the indel probabilities, and you can solve it. Of course, whether the missing data mechanism is ignorable or not is debatable. I think that for sequencing dropouts, this is a reasonable assumption. For heritable missing data, it's a little bit more subtle. Different models of heritable missing data will lead to missing data mechanisms that are ignorable or not ignorable. So another option is to model the heritable missing data process, but we prefer not to do that because we think that assuming ignorability requires less modeling assumptions, and it's a little bit simpler, but you could do that. And now what is perhaps my favorite part of this of this work, which is how to deal with the unknown ancestral states. And the way we do that is with this thing we call conservative maximum parsimony. This is, again, my favorite part of this. So let's talk about this. So even if you have an accurate reconstruction of the tree topology, we still do not know what the ancestral states are. Now, if there was no missing data in this assay, if we got to observe all the states, then there is in fact a unique, most parsimonious reconstruction of the ancestral states that is consistent with the CRISPR-Cas9 cutting process. So let me go over what the notation is here. So 0 is used to denote the uncut state. So this is the original state with the where CRISPR-Cas9 will identify and cut, and then other integers represent other indels that are produced when Cas9 cuts. So in this example, if you're looking for a most parsimonious reconstruction, then this is in fact unique, and it's this thing on the left. So there's no mutation supported on the first edge, then there's a mutation from 0 to 1 on this edge, then of course gets inherited by these two cells. And on the rightmost lineage, there's no mutation supported to this common ancestor, and then the mutation from 0 to 1 and from 0 to 2 happens. Now, if there's missing data, then that's where things get really tricky. So if there's missing data, the first issue is there's multiple most parsimonious reconstructions that are consistent with the CRISPR-Cas9 cutting process. So in this most minimal example, where we just have a tree over 2 leaves, so okay, the root is uncut. This leaf has an indel number 1, and this other leaf, the rightmost leaf, has missing data. So there's 2 evolutionary histories that are consistent with the observed data. The first one on top poses that a mutation happened from the root to this internal node, and the other one poses that the mutation happened on the way from the internal node to the leaf. So what do we do? So if we wanted to impede all the ancestral states, now we have to choose between these 2, basically. And if you run something like a variant of the Fitch algorithm, then it's going to select this one on the top. And what we propose to do is to not do this. What we propose to do is to only impute the ancestral states that are consistent across all valid most parsimonious reconstructions of the developmental history. So in this case, because this node, this internal node, has distinct state assignments under 2 different most parsimonious reconstructions, we will leave it unimputed and marginalize it out in the MLE. So this looks cool, but now there's a problem, because the only reason we were able to solve for the MLE was because, provided that you had all the ancestral states, the likelihood is really simple, and it does not depend on the indel distribution and so on. Well, what's interesting about this is, well, first of all, it's easy to compute the conservative most parsimonious reconstruction in linear time, but also the likelihood under the conservative most parsimonious reconstruction, or just CMP for sure, is easy to compute, just as easy as for the most parsimonious one, essentially. Because what's going to happen is that you're never going to have an internal state that's actually acting as a confounder. So you don't really need to marginalize it out. So, okay, now I'm going to show you some of my favorite results, which is that this works really well. And so what we've done here is we've really pushed our branch length estimator to the limit by simulating 100,000 characters down a tree. And just for reference, usually in these lineage tracing assays, nowadays we get, let's say, 50 characters, so 50 target sites. And hopefully this will continue to increase with time as these technologies improve. But here we really wanted to expose the bias of this estimator. And so we just cranked up the number of characters to 100,000 such that the variance tends to 0. And now whatever remains, the risk of this estimator is now just governed by the bias. So here's the ground truth tree. And now let me show you what happens when we fit the MLE when we have access to the ground truth ancestral states. So as you suspect, probably it's going to be really good. And indeed, here it is. It's basically identical because nothing is misspecified right here. So the branch length estimates are really, really good. Now let me show you what happens if you use maximum parsimony, so a variant of the Fitch algorithm, to impute the ancestral states. And you're going to get this. So as you can see, and as is well understood, so because maximum parsimony, and specifically the Fitch algorithm, tends to push mutations up the tree as much as it can. Now, a lot of the mutations have been pushed far up the tree, and the length of the branches at the leaves is now really short compared to what they should be. So now I'm just alternating back and forth. And so now I'm going to show you what happens if you use conservative maximum parsimony. So if you only impute the ancestral states that are supported across all most parsimonious reconstructions of this data, and this is this. And if I now toggle with the ground truth tree, you're going to see that, again, it's basically recovered. So I think this is really cool. This is my favorite part of this work. And so now let's, oh, okay, yes. Let me show you some of the more, some more quantitative results. But the reason this works, really the reason this is effective, is because CRISPR-Cas9 has a large state space. So the figures I've shown you right now correspond to the last row of this figure. So in the simulated data, there's a lot of details as to how exactly we've simulated the tree and how exactly we've simulated the lineage tracing data. But in this experiment, we've simulated 100 different indel states with a very skewed indel distribution, splined from real data. But there's 100 states, so representing the large diversity of indels that we observe in lineage tracing data. Now, if there were less states, so if we had a binary system, for example, where there was just the uncut state and one cut state, then you can see that naive maximum parsimony and conservative maximum parsimony still incur a lot of bias compared to having the ground truth ancestral states. Conservative maximum parsimony is still better. But the really cool thing is that as the state space goes larger and larger, the bias from maximum conservative parsimony goes down to 0, essentially, whereas naive maximum parsimony never recovers. So I think this is really, really cool.
Let's see. So we do have a question from the audience, Aaron McKenna, who you may know of. He says, I probably missed this, but in conservative maximum parsimony, how do you avoid propagating the unknown state all the way up to the root?
How do you avoid propagating the unknown state all the way up to the root? Well, we do have a lot of, well, I mean, there are going to be some internal states that are missing. I mean, it's not going to go all the way up to the root because we know that the root is 0. But
I think it might help. I think it would probably help if you would just describe what is the situation in which, okay, we're propagating this unknown state up, and then it stops at some point. What is the condition in which the unknown state stops?
Oh, well, I would need to, there's a slightly involved combinatorial characterization of how this happens. So yeah.
But wait, isn't it just when you have like multiple lineages that have data that's going up?
It's a little bit subtle. I wasn't gonna talk about it here, but hopefully in the preprint that we'll put out soon.
Yeah, really? Okay.
Yeah.
It's so subtle. Okay, I guess. Okay.
It's you don't like you can't just do it in one upward pass. Like you need to see what's going on higher up above you.
Wait, so how many passes do you need to do this conservative?
Two passes. First we do like the standard Fitch kind of thing.
Yeah.
And then we need to do one more upward pass to do this.
You can't just say like what happens with a Fitch set. Like we have the Fitch set and we have sort of something, the downward Fitch.
So it depends on, okay. It depends on, you need to look at the two lineages under you, and you need to see if there's a unique indel and if it's repeated across both or if it's just on one side. So yeah, I mean, what else can I add? I mean, yeah, it's a little bit subtle.
Okay. Maybe, and I think I'll, yes, I'll look forward to the preprint. Maybe I can just ask a related question that I was going to wait till the end, but here we are.
Oh yes, go ahead.
Branch length. So you have the point, like I'm just thinking about branch lengths and these unknown states. And I think the point is that you have lots of different of these targets. And so if you have missing data in some of them, that's okay. You estimate the branch length with the rest.
Yes. Yeah, exactly. So in fact, one of the characters might get cuts on the branch on the root. In that case, every single cell in your population is going to have that same indel. And so that is not really giving you a whole lot of information except for the branch length at the root, which you can see is probably not 0, certainly not 0, probably a little bit larger.
Great.
So yeah, all of this comes from the success of this comes from having many characters, and even then, this is still noisy thing. So yeah, now we're gonna show you some of the simulation results, and you're gonna see how as we crank up the missing data, things get worse and worse.
Okay, thanks for that question, Aaron, and keep them coming.
Yeah, keep them going, because I don't have I have more slides, but certainly not like infinitely more slides, such that I'll end up at like 10 p.m. So we should definitely, questions would be great too. So yes, this my favorite experiment. I love this. I think it's super cool. I think many of the things we talked about so far are relatively simple, if not trivial. But I think this is a little bit more nuanced. So I really, really like this. So now let's talk about how we evaluate this. And well, I've shown you, one of the neat ways in which we really push these estimators to the limits. And of course, the reason we can do this experiment is because the MLE is so scalable. Because really, the MLE just depends on the number of characters that get cut or that do not get cut on a branch. Those are sufficient statistics. So basically, the size of the optimization problem, the runtime, does not depend on the number of characters you have once you've computed the sufficient statistics, of course. So this is really nice. Now let me show you some results on more realistic regimes, what we do is, so first we simulate a single cell chronogram with its associated lineage tracing data. Then we subsample extant cells, and this is again something that we were talking about earlier, and this is a really important aspect of this problem, which is that we subsample things. And subsampling leaves a very distinctive signature on the tree. So you'll see that many of the branches near the leaves are very long. So if you were to naively think that this is exactly the cell population you're dealing with, it seems like cell division is slowing down. And that's not really what's happening. What's happening is you're subsampling, that's pruning away a lot of the branches and the leaves of the tree. So then we reconstruct the chronogram from lineage tracing data. We reconstruct the topology, we reconstruct the branch lengths. And if you pay close attention, you'll see that we have some branch lengths that are 0 here. So you would correctly guess that this is what you get out of the unregularized MLE. And finally, we evaluate the quality of the reconstruction using several metrics. So in this case, one of the simpler metrics to look at is what's happening with the time of the nodes in the tree, in the grand truth tree, and in the reconstructed tree. So simulating lineage tracing data is tricky. There's a lot of there's a lot of things that are going on. So the number of characters that we have, the amounts of missing data, how exactly missing data is modeled, the number of states, what's the state distribution, the mutation rates, are they the same, are they not the same? And no, in our simulations, we do not use IID rates, because we actually think this is a really important aspect of the problem. And this is one thing we're currently working on, which is extending this to deal with non-IID rates, which is not too hard. So let me show you some results. So we're going to look at this figure, and I'm going to hopefully walk you through this figure and show you what each thing means. So we have here a bunch of variants of our MLE. And what are those variants? So first of all, how much we regularize with pseudocounts. So lambda equals 0, the first 3 rows, there's no regularization. 0.1, which is a small amount of regularization, and 0.5, which is a larger amount of regularization. And then for each value of regularization, we can see what happens when we use ground truth ancestral states, when we use maximum parsimony or considerable maximum parsimony. And in this case, we're using the ground truth topology. So we are not reconstructing topologies at this point. I'll show this in the next slide, but here we have the ground truth tree, and we're just trying to estimate branch lengths. So again, to repeat this, we are varying 2 different axes, the first of which is how much regularization we use in the form of pseudocounts, and then the other axis is what is the ancestral states that are being used. And because we have the ground truth tree, and because we simulated this data, we know what the ground truth states are, and we can see how that is performing compared to the other things. And down here, we have the baselines. And here, the baseline that we're using is a really simple baseline where we just count the number of mutations on each edge, and then we extend all the branches so that it leads to an ultrametric tree, which is actually a pretty reasonable baseline on some tasks, because it has this implicit bias of creating long branches towards the leaf, which is really consistent with the subsampling process. So that's in red down here. And this is, again, the metric here is just how well the internal times of the nodes is predicted. And we're looking at the mean absolute error in this case. It's not the spurion correlation. We want to see what's going on with the absolute times. So things you will observe. So first of all, if you focus on the dimension which has to do with the ancestral states that are being used, you will see that using the ground truth states performs better than using either maximum parsimony or conservative maximum parsimony as expected. What's going on along this axis, which I haven't mentioned so far. So here we're varying the amount of missing data that there is in the experiment from 0%. So if the amount of missing data is 0, then we know that there's a unique most parsimonious reconstruction. So maximum parsimony or conservative maximum parsimony are the same thing. And they're not the same as ground truth states, but you can see that in this regime where there's no missing data, they do perform just as well as having the ground truth states. So, the way I come to think about this problem, it's really, the problem is not maximum parsimony on its own. The problem is maximum parsimony when there's missing data. If there was no missing data, I think maximum parsimony is a really fantastic assumption for the CRISPR-Cas9 process. But as you start cranking up the missing data, as I showed you before in our simulations, that's when maximum parsimony starts going wrong. And that's where you can see that using conservative maximum parsimony, there's a lot better than just using maximum parsimony. And the more missing data you have, the bigger the effect is. So suppose that you have like 40% missing data and you're using lambda 0.1, then naive maximum parsimony gets you a mean absolute error of here's 7.3, and ground truth states is 6, and conservative MP closes the gap like 2/3 of the way. It's 64. So I think this is really cool. So that's about the ancestral states choice. And then regarding regularization, you will see that using some amount of regularization also seems to help compared to no regularization at all. Now, okay, this looks really good. And how the problem is, in real life, we do not know what the topology is. We need to estimate that. So what happens when you estimate it? Well, when you estimate it, things get worse, and these benefits do diminish. quite a bit. So in blue, again, we have our original, our method when it's variance. Since we're now estimating the topology, there's no notion of ground truth states. And for this metric that we compute, that is based, again, on the time of the internal nodes. And it's a little bit more nuanced now that we do not know the ground truth topology. You'll observe that, again, using conservative maximum parsimony still does a little bit better than using naive maximum parsimony, and also that using some amount of regularization tends to help. But the results definitely, the improvements definitely diminish compared to the known topology. So I think the uncertainty in the topology is definitely, a really important part. And when you're doing all these downstream things, their performance is affected. You're, carrying over whatever error you had from the previous stage, and there's, quite a few stages here. So this is why, lineage tracing data is really tricky to work with. In this case, so here we're using one of the solvers from our package. It's the max cut solver based on a maximum cut algorithm, which tends to perform pretty well. Except that, of course, here we're just taking it to the limit as we increase missing data, and yeah, things do get worse. I talk a lot about scalability. I haven't really given numbers, so let me actually give you some numbers. So this takes around 1 minute to estimate branch lengths on a single core for 400 cells, which is what we've been using throughout all of these experiments. You will use that. So I haven't gone into all the exact details for how to compute the likelihood under a conservative, most parsimonious reconstruction of ancestral states, but the size of the computation graph is a little bit larger than for maximum parsimony. In fact, potentially in the worst case, it's quadratically. It depends quadratically on the number of leaves instead of linearly. But in practice, of course, we never observe worst cases, and it's just a little bit slower, and a little bit just like a 10-20% slower. And so, that's what we mean by scalability in this context. And of course, scalability is nice for many reasons. One of which is, and I haven't talked about this, but these estimates, these point estimates are not necessarily accurate. Depending on the quality of your lineage tracing data, it might vary a lot. And so having a scalable method means you can bootstrap your data. You can resample your characters, for example, and re-estimate either the topology or the branch length and see how reproducible your results are. So when you have a scalable method, you can bootstrap things and get some good idea of how good or bad things are performing. So one last thing I want to mention, which I think is also really important and a really nice aspect of this MLE, is that it can be really easily extended, because this boils down to a convex optimization problem. It means that it's trivial to extend in many different directions. So site rate variation. This is one that we're currently exploring. There's tons of work in statistical phylogenetics on how site rate variation matters and how to deal with it. For example, the Lee and Gaskell paper extending these models of amino acid evolution to deal with site rate variation. That's really popular nowadays. Another thing I haven't talked about is non-ultrametric trees. In all these simulations, we have ultrametric trees, which means that all the leaves have the same depth, because they come, the typical use case is you using this mouse model, you sacrifice your mouse, you harvest the cells, you sequence them. All the cells were sequenced at the same time. But there are other experiments where you're going to have longitudinal samples. So you're going to sample cells, let's say 1 month into the experiment, and then 2 months into the experiment. And so now you know that these cells were sampled at different times, but they come from the same population of cells. And extending the MLE to deal with this is trivial, because you just need to have a different constraint on the relative depth of each leaf. And another use case that is interesting is placing bounds on the time of the internal nodes of the tree. And this happens when you have a population of cells that diverges at some known time. And then you know that the lowest common ancestor between any pair of cells in these two populations is going to have a time that is bounded based on the time when these populations diverge. And all of these are linear constraints that you can just throw into the convex optimization problem really easily. And so it's really easy to extend to these use cases. And And these are things that we started to work on a new project, and all these things actually matter there. So hopefully at some time in the future I'll get to chat about that project, but it's really exciting because the MLE and its flexibility really allows us to handle all these other use cases that, depending on what you're doing, could be really, really hard to add to the model. So conclusions. So single cell chronograms are really important, but are very tricky to estimate. To reiterate, missing data is a huge problem here. And the fact that we also do not know what the ancestral states are, so we need to deal with that somehow. And so here, what we've done is we propose a fast, extensible, and convex maximum likelihood estimator, which exploits these things we've talked about, the conservative version of maximum parsimony. And we also deal with missing data by using the ignorability assumption. And then we've talked about how we propose regularizing the MLE with these very simple schemes that seem to work well. And finally, talked about how we evaluate this on extensive simulations. And really, the thing I most like about these simulations are this idea of just generating tons of characters down the tree. This is something that I don't think is very frequently done, perhaps because a lot of these methods can't really scale up, but provided that they do, really pushing them to the limit is really interesting because it reveals all the biases and it really reveals what's the important next thing to pursue and what is actually something that is not that important. So, for example, if you do this with if you now misspecify the model and add site rate variation, you will now observe that there is an important bias, and it leaves a very specific signature on the tree. So I think simulations are really cool, and not just the traditional simulations, but these simulations where you're taking your method to the limit by simulating tons of characters. So again, I'm very excited about this. We've started applying this to some really interesting data sets that have a lot of these caveats that the MLE can work with. So I'm really excited to see what's going to come out of that. But as for this work, and this is the first time I'm presenting this publicly, so that's very exciting. And there will be a manuscript online sometime soon where you can read more about the details. I think conservative maximum parsimony is a really cool idea. So yeah, hopefully if you're interested, you'll be able to check that out there. So that's all. It looks like we have like 5 more minutes or so for questions. So I'd love to take more questions.
Great, thank you very much. I'll start with a question by Anna Shukova. She's also following up on the conservative maximum parsimony. So you want to know, is it the same as Madison and Madison 2003 algorithm of 2 tree traversals, bottom up and top down, calculating sets of most parsimonious states that might be non-unique for each node?
I don't know. I haven't checked that one out, but I should. What's the name again?
This is Madison and Madison.
Okay. Madison and Madison. Yeah, okay. 2000 what? 2003? Sorry. 2003. Okay, all right. Yeah, maybe we just reinvented the wheel.
Well, who knows? I mean, so I also think simulations are awesome, but I'm curious a little bit more. Can you say a little bit more about how the simulations are set up and what are some of the weirdnesses that show up in them? I mean, I think you mentioned, like, it went by really fast, but you said something about non-IID rates, and I was curious about that.
Site rate variation. So, right. So in our model in the MLE, we're assuming that all the sites are cut at the same rate. But in practice, this is not the case in real data because these fake genes, these barcodes, get inserted into the genome at different locations, and they might be more or less exposed to CRISPR-Cas9 cutting. So some of them will get cut at a lower rate, some of them will get cut at a higher rate. So in the simulations, what we do is we use a different rate for each site, and the rates that we used are splined from estimates, from moment-matched estimates on real data. So there's quite a bit of variation among the rates. And yeah, it would have been nice if I had included that figure, but I don't have it, so I can't show it to you. But in these simulations, we do not assume that these sites are cut at the same rate. So yeah.
So I mean, I don't know if the technology's improved or something, but I remember when we were working with this data, there was all sorts of terrible things could happen where you would have like one cut and another cut, and maybe that would cut out everything in between those two cuts. Or like you would have some cuts that like erase the signal from a previous one. And so, I mean, even like, how do you even think about, is that like, what is the parsimonious scenario that includes all of that universe of possible?
Yeah, that's the Gestalt technology, right? I'm not wrong.
Yeah.
Yeah. So the lineage tracing data we work with, these barcodes have 3 sites. I think the Gestalt technology has like 7 sites. So definitely, the longer these barcode gets, you can get these double resection events that just wipe away a lot of the intervening target sites. In our data, I think this is definitely something to probe deeper into. But so far, what we've done is we do not model this, and yeah, I mean, I think. Yeah, I think that this is something where I like to dig more into and see how much of a problem this is. But I think the idea is with these shorter barcodes, these kinds of crazier, heritable. Well, it's not necessarily it's not heritable missing data. It's actually it's creating an indel that spans the whole region. So it wouldn't even be a missing data mechanism. This is a different thing, right? This is
Yeah.
These are correlated indel states, basically. I think
That's fine. It gets even. I think that you can like make it even more complicated, because it seems like it seems at least conceivable that the rate of cutting would correspond to something about the developmental state of the cell.
It could also be a function of time, and I agree with that. I think it's tricky, and that's why I think it's really important to be able to get bootstrap estimates, for example, and be able to to run these methods on to resample the characters and see how things are performing.
But more importantly is just like throwing all this weird stuff potentially. If there's evidence of it into simulation.
Right, into the simulation. Yeah.
There's also
agreed. Yes.
Even something like, okay, if one barcode is cut, does that modify the rate of cutting of the other barcode? I don't know.
Yeah.
Maybe.
And I think I think it well, in some of these earlier papers where kind of these technologies get developed and so on, I think that there are some studies in trying to understand how long these barcodes can record and so on. But I think doing research on understanding to what extent assumptions that we make about this process are accurate could be really interesting. I don't know if there's a whole lot of these. Maybe there's a temptation to actually work with real data and discover new biology instead of trying to understand in which ways these things are misspecified. So I think all these things are important, and there might be even more things we do not know about. So all of that is important, and all I can say is yes, you're right about all these things, and I'm very aware of them. And so that's whenever I look at these trees, I try to be as careful as I can.
Yeah. So I'm actually, this is great that you have this slide up, because I'm just personally interested in how do you incorporate the ultrametricity assumption in your optimization problem?
Yeah, so the optimization problem is phrased, so the variables of this optimization problem are not the branch length, they're actually the times of the nodes in the tree.
Yeah.
So branch lengths are just an affine transformation of this thing. So all we do is we set the root to have a time of 0, and then we set all the leaves to have the same time, which are equality constraints between the variables. So that's it.
Cool. With respect to your pseudocounts and so on, it sort of makes me think like, Would it be possible to do MAP estimation with where you take a prior on what the branch length is? Would that still be convex?
Yeah. Yeah, you can do it. You have to be careful with what prior you put on the branch lengths. If you if you put a birth process prior, you're still gonna get some of these collapsing issues. And I don't think I put that on the manuscript, but depending on the prior that you put, it doesn't actually fix the problem. I think if you're doing map estimation, you're still gonna run into these issues. You need to be yeah, I tried with many things. I tried with L2 regularizers. I tried with all sorts of things. And I didn't mention this because, I think the MLE is fantastic, but we also implemented a Bayesian estimator based on a subsampled birth process. And it doesn't have the issue of these edges collapsing to zero. So I liked the trees it was producing generally, but it just does not, like scaling that. First of all, scaling it up so that it runs faster, and also incorporating all these different aspects, such as the trees that are not ultrametric and placing bounds on the internal node. That just becomes dealing with site rate variation. It just doesn't scale up as well. But I think Bayesian methods are also a really cool way of dealing with these things. I think that they are just not as extensible, and they are significantly slower. But yeah, priors, map estimates, Bayesian estimates, other ways to go about this.
Cool. Awesome. Well, we're at time. That was a really fun and clear talk. Thank you so much.
Yeah, thanks everyone for listening. And I'm very excited to having able to present this. Hopefully the manuscript will be up soon.
Yeah, looking forward to it.